Okay, so there have been a lot of questions about DNA replication, and maybe it wasn't covered quite so well in lab, so I was just going to do a quick video for you. So I've written up here, this is my DNA strand. It's going on the top is 5' down to 3', and on the bottom it's going 3' to 5'. Okay, so if this is DNA replication, and obviously I'm not going to draw all the letters, but there's our DNA strand. So what happens originally is somewhere along the DNA strand, an enzyme opens it up. Okay, so we've got an enzyme that's opening it up. So here we have it, and we're going to call it helicase. So that's the enzyme that breaks the helix. So we label it helicase. Okay, so it's unzipping this DNA strand going this way. Well, new DNA is built 5' to 3'. So if we're building 5' to 3', then we're going to build along this bottom strand that's running 3' to 5'. The problem is that the enzyme that builds DNA is called DNA polymerase, and DNA polymerase, for some reason, it cannot lay down the first base. It can only add to an existing 3' end. So what we have to have is another enzyme that comes in, we're going to call it primase, and primase comes in and lays down an RNA primer. It's made out of RNA. RNA can be added, just like in RNA polymerase and transcription translation, you don't need a primer. It can add to nothing, so RNA polymerase comes in and adds. And then we have our DNA polymerase enzyme. So here's our DNA polymerase, and it's specifically DNA polymerase number 3. Okay, and DNA polymerase number 3 is going to come in, and it's going to copy the DNA. So it's just cooking along, going from 5' to 3', and it's following the helicase unzipper. Okay, so it's going to go along. But let's look at the other strand. So the other strand, because it's unzipping this way, the other strand originally probably started over here with a primer, and then our DNA polymerase came in and copied the DNA like this, going in the other direction, right? Because the strands are anti-parallel. But then the helicase opens it up more. So oops, it's got to go in and it's got to start it again with another primer, and then it's going to continue this way. Boom, boom, boom. Okay, going in that direction. And then it opens it again. So it's got to start another primer, right, like that, right? And keep going in this direction. Now watch, because it can only add to an existing 3' end, it actually can't make this last little gap right there, because this is adding to the wrong end. It can only add to that end. And so you end up with these little gaps. And so this bottom strand that's going just continuously is called the leading strand. Leading strand. Whereas this other strand that's having to restart over and over again is called the lagging strand. I'm sorry, these markers are not great. Just give me 1 second. Okay, so it's called the lagging strand, and it makes what we call Okazaki fragments. Okay, and that's these little fragments of DNA. So the other thing that happens is originally when the DNA strand is opened up, the single-stranded DNA is delicate, and so as a consequence, you get these little single-stranded binding proteins that grab onto the DNA just to make sure it doesn't break while it's open and single-stranded. Single-stranded binding proteins. Okay, so here we have our DNA being copied, but the problem is that we have little bits of RNA that are still in the DNA. And so we have another DNA polymerase. We'll just draw it in blue. We have another DNA polymerase that comes in. Here's our DNA polymerase 1, okay? And it's going to come in, and it can add to that end, right? So it's going to come in, and it's going to add DNA, and it's going to erase the RNA that was there. And it's going to add. But again, that little gap right there, it can't add right there because, and let me zoom this in a little bit, because again, we're adding to the wrong end, so it can't add that gap right there. So it can replace the RNA primers, but it can't add the gap. So it's going to come in here too and replace, but it can't finish it. Okay, and it'll come in here and replace from the other end, but it can't finish it. And so we need one more enzyme that's going to come in, okay, and it's just going to glue those little pieces together, okay, and that enzyme is called ligase. For ligating, it's going to glue them together. So it will come in and finish the strands and ligate the whole thing together so we have continuous DNA, okay. Now, the other thing we have is, have you ever tried to unzip embroidery floss? When you do that, it like super winds underneath and gets really tangled. The same thing happens with DNA. And so we have an enzyme up here that grabs onto the DNA. It's called Topoisomerase. And its whole job is to split the DNA open, let it unwind, and then seal it back together so you don't get super winding of the DNA. And eventually, this is going to open all the way to the end, and it's going to copy the DNA. Now, let's look really closely at the end for just a minute. Let's see if I draw down here. Can you, over here, you can see. So let's pretend we're just going to take this and call it the end down here, okay? So at the end, where this is the 3' end and this is the 5' end, so our continuous strand, our leading strand, that enzyme's just going to be cooking along, copying, copying, copying all the way to the end. And then it's just going to fall off. On the other strand, we had a primer laid down, okay, and then the strand, and then the enzyme came in and copied it like this, going in the other direction, boom, boom, boom. Okay, but now when we have our DNA polymerase 1 try to come in and add, there's nothing over here to add to. And so as a consequence, it can't replace that primer. And we know that RNA is more degradable than DNA, so the RNA falls off eventually, which leaves this tiny little tail that's single-stranded. And so as a consequence, that breaks off. So each time you replicate your entire chromosome, a little bit on each end breaks off. And that's why we have telomeres. And the telomeres are all that junk on the end that allows it to lose a little bit each time instead of losing valuable information. Okay, so that's the way the DNA replication works. Now I saw a lot of confusion as I was just glancing at the posters between DNA replication and transcription and translation. They're similar processes, but for totally different things, right? So DNA replication happens during the S phase of the cell cycle in preparation for mitosis or meiosis. Transcription, that is just for making photocopies of recipes when we need to make the proteins. And so that, it happens in a similar fashion in that we unzip the DNA just in the spot where the gene is, but we don't have helicase and we don't have topoisomerases or single-stranded binding proteins or any of that. All we have is an RNA polymerase. Now remember, RNA polymerase is adding RNA. So you don't need a primer because RNA can be added to nothing. So all that happens for transcription is that the strand gets opened up and then the RNA polymerase sits down, reads the gene that it needs along the template strand, and then when it jumps off, it just binds back together. We're not making copies here. It just would bind back together. And I think I might actually draw it here in a minute, but I want to give you a close-up of this picture, okay, so that you can see it. I'm going to get my head out of the way. All right, so that's our DNA replication. Okay, so before I erase it, let me show you what would be if we were talking about transcription. We're going to get rid of all of these enzymes. We're not making a copy. We have no helicase. We have no topoisomerase, no single-stranded binding proteins. Okay, no leading strand or lagging strand. All we have is the piece of DNA, okay, that's simply been split open. And let's say that the coding strand, the gene, oops, let me get rid of all that up there too. Okay, so there's our coding strand. And this is our gene right here. And it's going 5' to 3', okay? And our DNA is simply split open by our big RNA polymerase enzyme. And the RNA polymerase enzyme is going to read this strand down here, which is the template. And this is the coding strand, where the gene is, okay? And so if we were to hypothetically put some letters in here, you know, A, U, G, it's not going to be a U, this is DNA. A, T, G, C, A, C, etc. Then down here you'd have T, A, C, G, T, G, etc. All right, so we're opening it up, and then our DNA polymerase, or our RNA polymerase enzyme it's going to sit down right here. So this is our RNA polymerase. And it's just going to copy, using RNA, it's going to copy this DNA strand. So when you see a T, you write an A. When you see an A, you write a U. When you see a C, you write a G, and so on. And so you can see that the mRNA, this right here is mRNA. The mRNA and the code are identical. They equal each other, okay? And then this mRNA, and one key trait for mRNA is it should start with AUG, right? That's your methionine codon, CAC. So there's our copy. When it comes in into the ribosomes, then our transfer RNA comes in with an anticodon. So it's going to be UAC, GUG, and these are each going to be their own little tRNAs. This one's going to carry methionine. This is going to carry a different one. I don't even know, and I'm not going to look it up. Okay, and so you can see that the right over here, the anticodons that are right here. So these are the anticodons. They look the same as the template strand, whereas the codons of the mRNA look the same as the coding strand. Okay. So that's what's happening, and then as soon as it's done making that copy, that falls out, and this DNA closes back up. And you don't have it hanging open like that. All right, I hope that made sense. Enjoy.